Dzień dobry Państwu. Dzień dobry, nazywam się Adam Szewczyk i będę pełnić dzisiaj rolę tak zwanego technicznego hosta naszego spotkania, czyli będę odpowiadał za stronę techniczną tej transmisji. Po raz pierwszy Polskie Towarzystwo Biochemiczne realizuje transmisję wykładu parnasowskiego przy użyciu platformy Zoom Webinar, a jednocześnie na koncie facebookowym Polskiego Towarzystwa Biochemicznego mamy live streaming, także jesteśmy obecni w internecie. Ta platforma umożliwia zadawanie pytań na końcu naszego wykładu i będziemy chcieli tą platformę wykorzystywać przynajmniej przez najbliższe miesiące do komunikacji różnych treści naukowych do społeczności biochemicznej. Plan dzisiejszego spotkania jest następujący, że przed wykładem pani profesor Magdy Konarskiej wstęp i laudację wygłosi prezes Polskiego Towarzystwa Biochemicznego profesor Andrzej Legocki, później będzie wykład i po wykładzie spróbujemy wykorzystać formułę Q&A zadawania pytań do profesor Konarskiej. I to od strony technicznej wszystko i teraz poproszę pana profesora Andrzeja Legockiego, prezesa Polskiego Towarzystwa Biochemicznego o zabranie głosu. Andrzeju, bardzo proszę.
Bardzo dziękuję, Adamie. Szanowni Państwo, corocznie przyznawana nagroda imienia Jakuba Karola Parnasa, Polskiego Towarzystwa Biochemicznego, nagroda za najlepszą pracę biochemiczną wykonaną w polskim laboratorium przez polskich biochemików, ma długą, bo już prawie 60-letnią tradycję. Nagroda ta od początku była uznawana i taką pozostaje za najbardziej prestiżowe wyróżnienie naszego towarzystwa. Z wielką przyjemnością chciałbym przedstawić Państwu profesor Magdę Konarską, która wystąpi dzisiaj z wykładem parnasowskim, który jest elementem tej nagrody. Pani profesor wystąpi w imieniu zespołu, któremu nagroda ta została w tym roku przyznana. Oprócz liderki tego zespołu, Magdy Konarskiej, w skład tego zespołu wchodzą Katarzyna Eismont, Katarzyna Matylla Kulińska, Agata Jaskulska i Marcin Magnus. 9-osobowej komisji, która rekomendowała zarządowi głównemu Towarzystwa nagrodzonych autorów i ich pracę, przewodniczył profesor Andrzej Dżubej. Profesor Magda Konarska wywodzi się ze środowiska warszawskiego, z którym od początku swojej drogi edukacyjnej i naukowej jest związana i gdzie uzyskała wszystkie stopnie naukowe. Była związana z Instytutem Biochemii i Biofizyki Polskiej Akademii Nauk, w którym wykonała pracę doktorską pod kierunkiem Witka Filipowicza w pracowni biosyntezy białka, którą wtedy kierował profesor Przemysław Szafrański. Obok Witka i Magdy z pracownią tą związani byli m.in. Piotr Chomczyński, Włodzimierz Zagórski i Jadwiga Wisia-Chroboczek. W roku 1985, szanowni Państwo, Magda Konarska otrzymała nagrodę imienia Jakuba Karola Parnasa. Jest więc jedyną dwukrotną laureatką tej nagrody. Magda Konarska posiada imponujący rekord staży i wizyt naukowych, zagranicznych. Pracowała w znakomitych miejscach i współpracowała ze znakomitymi uczonymi. Wymienię tutaj Hansa Grossa w Würzburgu w początkach jej drogi naukowej Później wielką renomę zdobyła w czasie 5-letniej pracy i współpracy w MIT z pracownią Filipa Sharpa, a później przez ponad ćwierć wieku, Magdo, nie wypominam ci tych lat, ale jestem wraz z tobą i z naszym towarzystwem z twojej działalności bardzo dumny. Przez ponad ćwierć wieku związana była z Uniwersytetem Rockefellera w Nowym Jorku, w którym zajmowała prestiżową pozycję lidera zespołu i kierownika pracowni biologii molekularnej. Problematyka badawcza Magdy Konarskiej zawsze była wyjątkowo zwarta i skupiona wokół zagadnień związanych z udziałem RNA w różnych procesach komórkowych, takich jak mechanizm ligacji RNA u eukariotów, to wcześniej, czy mechanizm splicingu jądrowych, prekursorowych mRNA i w ostatnich latach budowa spliceosomu, w szczególności rearanżację w jego centrum katalitycznym. Magda Konarska wybrana została do wielu najbardziej prestiżowych instytucji polskich i międzynarodowych. Wymienię tutaj Polską Akademię Nauk, EMBO, Europejską Organizację Biologii Molekularnej oraz Akademia Europea. Obecnie wraz z Agnieszką Hacińską kierować będzie nowym, będącym ciągle chyba w trakcie organizacji Centrum Badawczym Polskiej Akademii Nauk w Jabłonnej. Podstawą finansową tego centrum, Instytutu Międzynarodowego Mechanizmów i Maszyn Molekularnych, utworzyły fundusze Międzynarodowej Agendy Badawczej MAB, które to fundusze obie uczone uzyskały. Magdo, the screen is yours.
Thank you, thank you, thank you. Thank you very much for this wonderful introduction. And thank you to you and to the entire Polish Biochemical Society for this wonderful award. I actually didn't realize that I was the only one who got this award 2 times. But I was thinking that in conveying my gratitude, I am really very happy to do that, not only in my own name, but also in the name of all my coworkers, because I know what it meant to me when I was in their position all those years ago. So I hope that for them, this wonderful start in their scientific careers will lead them to wonderful things in the future. I have to say that when I first was told about this great recognition, I was initially troubled for a moment because I wasn't sure how will I justify being recognized by the Biochemical Society for the work that is, as you will see, done primarily using genetic methods in yeast. But I hope I actually solved this problem for myself, and I hope I will convince you that it is okay in the end, because what we are doing is we are interested in basic fundamental biochemical questions. Basically, it is pure enzymology what we do. It's molecular enzymology of an incredibly complicated enzyme. It's called the spliceosome. And it's because of its complexity, I decided a long time ago that the only way to really address the mechanism of this enzyme is to actually study it in vivo, and therefore using methods of yeast genetics. And so you will see, and I hope I will convince you that the basic work then is purely biochemical at heart. So maybe with that, I will share my screen with slides. Just a moment. Okay, I hope you now see that. And so today I want to talk about well, the catalytic core of the spliceosome. But before we get to that, maybe I will start by reminding everybody what splicing really is. So it's a process that is universally present in all eukaryotes. And it consists of, so the basic idea is to remove from the primary transcript of pre-mRNAs in the cell, to remove the intervening sequences, so-called introns. Oops, sorry. Yeah, that's going to be a problem. To remove the intervening sequence, so-called intron, and splice together, join together the remaining coding sequences, so-called exons, to produce mature mRNA, which is then functional for translation of proteins. Splicing, although it's a single process, it in fact consists of two separate reactions. We call them steps of splicing. Oh, sorry about that. And in the first of these two reactions, a nucleotide in the intron attacks a junction between the 5' exon and the intron. And as a result of this attack, the 5' exon is cleaved off, and this nucleotide is attached to the 5' end of the intron, forming this very characteristic lariat structure, lariat intermediate. So this is the first reaction of splicing. In the second reaction, subsequent reaction, it is the 5' exon, which was cleaved off in the first reaction, which now attacks the junction between the intron and the other exon, the 3' exon. And again, it cleaves off, this time cleaves off this lariat intron and joins together with the other exon to form mature mRNA. So these are two relatively simple consecutive reactions that are catalyzed by a single enzyme, a single catalytic center. However, the enzyme is remarkably complex. It is called the spliceosome. It is composed of 5 subunits. We call them SNRPs. Each SNRP contains one piece of RNA, snRNA, and so we have U1, U2, U4, U5, and U6 RNAs, snRNAs. But each of them is associated with a number of proteins. And in addition, there are other proteins, transacting factors that join in and leave this whole complicated process of putting the spliceosome together. Now, remember that the spliceosome assembles on each intron de novo. So this whole process has to be repeated for each individual intron that is subjected to splicing. And so we have these individual steps, very complicated assembly with SNRPs coming and leaving and other proteins coming and leaving. And finally, we enter into the catalytic phase of the process, which is the main topic of my interest in the lab. But the complexity of these enzymes then is not only due to the fact that its composition is so amazing, because in total, in the spliceosome in yeast, which is a simpler, smaller spliceosome, there is about 100 different proteins involved in it. In us, in human cells, the spliceosome count of proteins involved in splicing is way above 300. I think it's 350 or more. So you can see that the level of complexity of this gigantic enzyme. It is perhaps the largest and most complex enzyme in the cell. And then finally, after this complicated process of spliceosome assembly, we enter splicing catalysis. Here, still some components are leaving and joining. But in addition to that, there are important conformational changes that need to take place. And so each of these individual steps in this cycle represents a transition from one complex to the next. And these transitions are facilitated by a dedicated class of enzymes, ATPases, which facilitate progression of this whole process. And so, for example, then, if we look at the catalytic phase here, and focus at least, for example, at this enzyme PRP16, it controls the transition from the first step catalytic conformation, which supports this reaction, first reaction of splicing. And then only upon PRP16 action, this complex makes a transition to another conformation, which leads to the second step. So the second reaction can take place. Using genetics, we can then mutate, this is complicated. We can use a mutant form of this enzyme, which will result in accumulation of these complexes and improved first step catalysis. However, by contrast, of course, the second step catalysis will be inhibited because these complexes have hard time exiting the first step catalytic conformation. Okay, so far everything that I showed you and told you about has been figured out primarily based on biochemical and genetic studies over the more than 40 years of splicing since the discovery of splicing. However, in recent years, there was a major revolution in the field with new structural information as these multiple complexes, multiple forms of spliceosomes were solved by cryo-EM. And this was done by 3 labs headed by Kiyoshi Nagai, Yigong Shi, and Reinhard Luhrmann. And so you can imagine that now with this amazing information, detailed information about interactions and changes between these different complexes, this brings us to an entirely different level of analysis. And so, of course, since I'm always have been mostly interested in catalysis per se, the first thing was to look at the catalytic center. And here these structures fully confirmed the models that were predicted prior to the structural analysis. And this is primarily based on genetics, that the catalytic center of the spliceosome is built primarily of RNA. So this is basically, the spliceosome is a ribozyme. So RNA structures form the catalytic center. However, this catalytic core, then RNA catalytic core, operates in the context of these hundreds of proteins. So the spliceosome as a whole is a ribonucleoprotein ribozyme. When we look more closely at the catalytic center of these RNAs, you can see, so here is the structure from the cryo-EM structures. Here is its simplified representation in 2D. In all throughout my talk, I will be showing you U6 snRNA, one very important component of this catalytic center. It always will be in red. And U2 snRNA, another important component, will always be in green. Here we also have their interaction with the intron, exon, and U5 snRNA. So this is the complete catalytic center of the spliceosome. But looking more specifically, then this catalytic center includes a very important element, so-called the catalytic triplex. So this is a structure which is formed by 3 strands of RNA, 2 of them provided by U6, and the third provided by U2. And so these are 3 consecutive triples that clamp this conformation into the catalytic structure. And importantly, they also coordinate metal ions for catalysis. So basically we look here at the active site of the spliceosome. And as you can see, it is formed entirely by RNA. So naturally, when these structures of cryo-EM structures of different spliceosomes were formed, we were immediately drawn to the question, so what happens to the catalytic center at these different stages of spliceosome rearrangements that are so characteristic of this whole process? And to my big surprise well, I was quite perplexed seeing that in fact, the catalytic center doesn't change in these structures. Here I'm showing you superimposition of U6 snRNAs in all these different complexes. And you can see that it's fully superimposable. U6 doesn't change. And the same applies to other elements here. So this was really surprising and troubling. Why? Because if we look at the fundamental essence of this reaction, what it is, well, I told you that the splicing catalysis involves two separate reactions. So we have a single catalytic center that needs to bind substrates for the first step, compress and induce catalytic conformation that carries out splicing catalysis, then needs to open and release substrates, needs to allow for repositioning of substrates such that substrates for the second step enter the catalytic center, and then carry out second step catalysis and finally release the products. And so this was not impossible, but very difficult for me to imagine how could this happen without any change on the part of the catalytic center. Secondly, this PRP16 enzyme, the ATPase that promotes this transition from the first to second step. Mutants of this enzyme cause serious defects. So the strains barely grow, don't tolerate low temperatures. So again, how come if the catalytic center doesn't change? Now, for these two reasons, well, I could still come up with some explanations, maybe. But the third fact that was difficult to reconcile was that we knew at that stage, we knew of 2 or maybe 3 mutants in U6 RNA, a component of the catalytic center that promoted or improved, strongly improved either the first or the second step catalysis. And that is very hard to explain if the catalytic center doesn't change. So because of that, we actually considered an alternative hypothesis, and this is that the spliceosome or the structure at the catalytic center does indeed change, toggling between a more stable intermediate structure, which is not capable of catalysis, and less stable transient catalytic conformation. And structural analysis by cryo-EM could capture only these intermediate structures, however, did not capture the actual catalytic state of the catalytic center. And so to ask if this hypothesis is correct, Kasia Eismont, a graduate student in the lab, decided to look more closely at U6 RNA and introduce mutants in this catalytic center or U6 component of the catalytic center and ask how will they affect behavior of PRP16. So remember, this is the enzyme that promotes this transition, exit out of the first step and entry into the second step. When mutated, this is inhibited, these complexes accumulate, first step catalysis is improved, but the second step is inhibited. And this results in growth defects, temperature, cold temperature sensitivity. And so Kasia was asking if any of these mutants would change this phenotype of these strains. And she made many mutants. I will start by showing you just a few examples and then conclude more broadly. So she started with, for example, this mutant here, a single point mutation in this lower segment of U6 ISL. This disrupts this base pairing in this lower stem and results in worsening of defects. So now these strains don't grow even at 20 degrees. And so we conclude that they further stabilize the first step catalytic conformation. And in fact, they improve catalysis, first step catalysis. Similarly, another mutant on the other side of this stem does exactly the same. It also further inhibits growth of PRP16 mutants, further stabilizes first step catalytic conformation. However, when Kasia combined these two to restore this base pair, now the strains grow perfectly fine. They in fact grow better than the original wild type strain. And we think that this is because this stability of this new CG base pair is higher than of the original wild type UA. And so we conclude that the stability of this lower ISL segment is critical for the behavior of at this transition between first and second step. So unstable lower stem structures promote catalysis, first step catalytic conformation, whereas stabilized stem loop structure in this lower segment improves exit out of the catalytic conformation. So here, this is a full list of mutants that Kasia made and tested. This was a gigantic effort. And to simplify it, I will show you a schematic which summarizes these results. So whenever Kasia disrupted or mutated elements of the catalytic triplex or of elements of this upper segment in U6, this always resulted in inhibition of catalysis and improvement of the transition to the second step. However, this element, the lower stem of ISL, behaves in the opposite way. That is, here, destabilization or disruption of this stem, making it more flexible, improves catalysis. So in other words, we conclude that catalytic conformation requires stable catalytic triplex and flexible, unstable lower stem of U6 ISL. To confirm beyond any doubt that in fact the spliceosome or the catalytic center of the spliceosome does undergo repetitive conformational changes at these transitions between the reactions, and that this change, maybe I should first explain. So we think that this structure changes between the intermediate structure that has a stable lower stem and the catalytic conformation that has a flexible lower stem. And to make sure that this is really how the catalytic center works, Kasia made another mutant in which she artificially hyperstabilized this non-active, non-catalytic structure by changing this single mutant this single nucleotide to an A. She forced base pairing between this new position and this U residue, which normally is bulged out and is involved in formation of the catalytic triplex. Now that there is this perfect base pair here, this whole structure has a very hard time adopting this catalytic conformation involving this formation of this triple UCG. And so this strain with this mutation is actually very sick. It's barely viable. However, Kasia could make it, could improve its growth greatly by introducing mutations that destabilize lower stems. Here is one such example. And you can see that now in the presence of this mutation here, this strain restores perfect growth. Another way to fix this difficulty of this stringent and strict and inflexible stem loop structure is to improve and further strengthen the catalytic triplex. So in the normal wild type situation, this UCG triplet is semi-stable, it's non-canonical, it is okay, but in the presence of this mutation, this is not sufficient to support catalytic conformation. And therefore this strain, again, just like here, it doesn't grow basically. However, when Kasia strengthened this triplet by introducing a canonical perfect triplet UAU, this greatly improved growth of this strain. And so basically these experiments confirm beyond any doubt that the two possible conformations existing at the catalytic center involve an intermediate non-catalytic conformation that involves stable pairing in U6 snRNA, or a catalytic conformation that involves flexible lower stem of U6-ISL and a stable catalytic triplex. So this then confirmed our original hypothesis that in fact the catalytic center toggles between these structures. And in fact, we believe that this structure, or at least similar to this structure, is then repeated again during the second step of splicing. And so we identified again a new element of the catalytic center, which is this flexible element at the lower segment of U6-ISL. So this makes this element particularly important. This is a new thing that we didn't know about. To learn more about it, Marcin Magnus, who is a postdoc expert in bioinformatics, who came to the lab to learn methods of molecular genetics. And he looked at U6-ISL in all eukaryotes, across all eukaryotic organisms. And he found that, strikingly, it's the upper segment of U6-ISL that is highly conserved. And it is highly conserved not only in all U6 molecules in all organisms, but also in U6-like molecules, U6 attack molecules that operate in a similar kind of a mirror image spliceosome, so-called minor spliceosome. So the sequences are different, but the stability of this upper element is absolutely conserved across all eukaryotes. Furthermore, this also is the case for group 2 introns. So remember, group 2 introns, you may not know, but these are RNA molecules that fold and form the catalytic center that catalyzes their own excision. And they form essentially equivalent structures, and the domain that is equivalent to this U6-ISL has the same feature, that the upper segment is highly conserved. However, the lower segment in all these cases is significantly less conserved. The next thing was that Marcin looked at stability of these two segments, and we start with the upper segment of ISL. These sequences are basically these segments in all U6 attack, and group 2 introns show essentially equivalent stability in all organisms. However, by contrast, stability of the lower stem is different, and most importantly, it is always less stable. So the lower stem is always less stable than the upper stem. So with no exception, this is less stable than the equivalent upper segment in U6 attack, or group 2 introns. Now, here there is an interesting thing, because this is yeast, Saccharomyces cerevisiae, our model system. And this is an extreme case where the stability of this upper and lower segment is very similar to each other. However, when Marcin analyzed stability of these mutant forms that Kasia analyzed in this work, they destabilize this element, and the stability of these mutant forms is now fully comparable to an average lower stem of U6 in other U6 or U6 attack, and so on. And this is an interesting case because this lowering stability of lower stem of U6 is to some extent it correlates with a reduced fidelity of splicing. So all these other organisms, except for yeast, can support extensive alternative splicing, that is splicing where different sites within a single molecule can be used as splice signals and processed by the spliceosome. In most cases, these alternative splice signals are suboptimal. And in yeast, they would not be tolerated by the strict and high fidelity spliceosome that is characteristic of the yeast structure. However, lowering of the stability of this element allows for tolerating all kinds of suboptimal splice signals and therefore resulting in alternative splicing, which in yeast, basically alternative splicing is not detected. So together then, this offers a very interesting new look at what is going on with spliceosomes in different organisms. When we take a catalytic center like the one in yeast, where the relatively speaking, this lower segment of U6 ISL is rather stable, much more stable than the equivalent structure in higher eukaryotes. This results in spliceosomes or catalytic centers that have hard time entering into the catalytic conformation, and therefore they will tolerate only perfect signals in their substrates, meaning that the fidelity of splicing in this case is very high. That is, suboptimal, weird, not perfect substrates will not be processed by such catalytic centers. However, with destabilized, more flexible structure of this element in higher eukaryotes, the spliceosome can more readily enter into catalytic conformation, and thus it will accept all kinds of even suboptimal substrates, therefore reducing splicing fidelity, but opening the possibility to alternative splicing. And this is really fascinating and may have very important consequences, because that means that if we change the sequence in this region of U6, we can introduce a new features, catalytic specificity of such spliceosomes. And this is interesting because it is only in yeast, Saccharomyces cerevisiae, that we have a single copy of each RNA, and every spliceosome has the same exact copy of U6 RNA in its spliceosome. However, in other organisms, and in particular in humans, there are multiple loci coding for U6 as well as other snRNAs, and these and thus, at least theoretically, opening the possibility that we might be dealing with multiple forms of spliceosomes in our cells, depending on what U6 RNA might be expressed. Now, granted, all these other loci of U6 and other snRNAs are called pseudogenes, and they are thought not to be expressed. However, first of all, this problem has not been studied very extensively. And secondly, at least some of these so-called pseudogenes are in fact expressed, and this has been documented. So then it is possible to speculate that perhaps, at least under certain conditions in certain cell types, some of these variant copies of U6 might be expressed, incorporated into the spliceosome, and alter its behavior. And so here I'm showing you some of these variants. In green, there are deviations from this perfect, optimal, perfect, canonical U6 that typically is highly expressed in cells, in human cells. And here in those red boxes, I highlighted deviations in these variants from the wild type that correspond to this lower stem of ISL. And so you can look at it and very easily realize that, well, that means that if some of these sequences were to be expressed and incorporated into spliceosomes, we would have different spliceosomes with different properties of their catalytic center, thus allowing for these tailor-made spliceosomes that will be optimal for processing of this or that type of intron with particular suboptimal problems in their splicing signals. So this is, of course, just a speculation. This is something that I would love to study if we ever are allowed to go back to work. But for now, I wanted just to summarize and say that, in fact, this analysis using genetic tools did offer us a very interesting new glimpse into the behavior of the catalytic center of the spliceosome. We identified a new element, a flexible lower stem of ISL, which represents an important new element critical for splicing catalysis and catalytic conformation. And we believe that spliceosome really does toggle between these more stringent and stiff intermediate structures, which are not catalytic, and these flexible catalytic conformations. But thus, so far, only these structures could be captured in cryo-EM structures. So this is what I prepared for you today, and this is now the most important slide. I wanted to introduce the heroes of this work. First of all, Kasia Eismont, a graduate student in the lab and the first author of this awarded paper. She was helped by Kasia Matyla Kulińska here, a postdoc in the lab, as well as Agata Jaskulska, another graduate student. And finally, Marcin Magnus, postdoc bioinformatics guru who helped us with analysis of U6 structures. And all of these co-authors, as well as the rest of my lab, are greatly grateful to the Polish Biochemical Society for this wonderful award. Thank you very much. And I think I will now take questions if there are any. Thank you very much.
Thank you very much indeed, Magda.
Now, concerning the questions, there is a place down on the screen, Q&A, and that's the way of questioning. Do you see the question, Magda?
Actually, now I can see the question. Yes. So the first question is from Zosia Szwajkowska-Kulińska with the question: do yeast cells with mutated U6 ISL perform alternative splicing? Well, this is a key question. What we can say is that they definitely tolerate suboptimal substrates. So now they can splice all kinds of mutants, intron mutants, that otherwise would not be spliced by normal wild type sequence. However, when we looked at the full spectrum of splicing patterns in the cell, we were not able to detect dramatic changes in alternative splicing. There are some minor changes, however, not all that much. Now, this could be because we analyzed this in the context of cells that were, in the context of RNA that was selected as poly A+. And thus, most likely, we did not capture altered splicing events that were not leading to stable polyadenylated RNAs properly exported to the cytoplasm. So this is an open question that we would like to be able to come back to and analyze at greater detail, because I suspect that it should happen. It's just that the way that the experiment has been done so far did not allow us for a conclusive, definitive answer.
Okay, and we have the second question on the Q&A.
Yes, thank you. Kasia Knop is asking, do you know anything about conservation of U6 snRNAs in plants? So let's start with that. So this is, plants, in fact, again, they fully match this pattern that I showed you earlier. Maybe we can go back to this picture here. So plants, in fact, this element in plants is very similar to humans. And so basically this sequence is unchanged. Now you have to remember that U6 in general is one of the most highly conserved sequences, definitely in the spliceosome, but in general, it is very, very highly conserved. And so no, plants in this element, it behaves just like some of these other eukaryotic organisms. There is nothing particularly different about this element between plants and humans. Now, the second part of this question is there any indication about the influence of RNA modifications on U6 snRNA structure in yeast? Now, that is a great question. And yes, in general, yes, this is poorly studied, but there are examples where modifications, so I know of pseudo-U, pseudo-uridylation events that actually alter behavior of U6. This is definitely something that should be studied at much greater detail, and I would love to learn more about it, because I'm convinced that yes, there must be more. Also, it is quite possible that there are other modifications, for example, even transient modifications that were not even studied, and we don't know what effect they might have.
Okay, that's done. Next question from Joanna Zawadzka-Pankał.
Yes. Are alterations in snRNAs linked to any human cancer? Well, not to my knowledge, because again, because all these variants that are that exist are treated as pseudogenes, and nobody looked at the expression of these variants in cancer. I mean, this is essentially the question that I wanted to ask. How does it change in cancer cells at different developmental stages? You know, what is happening there? I suspect that there may be significant changes. We have not been able to address that experimentally, or well, that's because of the peculiar state of my lab at this point. But these are exactly the questions that I would love to address. And I am convinced that the answer must be yes, that there has to be a change there.
Okay, the next question from person HP.
Mm-hmm. If there is correlation between splicing and production of microRNA. Oh, so here I would so in yeast, where we analyze these processes, there are no microRNAs, and therefore this is not applicable to our system. But in general, I would refer you to other people studying it more closely, in particular, Zosia Szwejkowska-Kolińska. Another question is from Tomasz Stępkowski. Do you believe that in light of our results, we will have new tools, methods for biological engineering at post-transcriptional level, for example, to precisely regulate alternative splicing by decreasing the levels of particular snRNAs or some other approach? Well, you know, that's exactly the direction that opens up now. I think that this sooner or later, this will be tried, because this is such an obvious direction. However, remember also that it is not enough to, let's say, express a new type of RNA, U6 RNA in this, I don't know, cancer cell or this or that, because if it is, so I don't know how to make it specific to a particular type of introns at this point, right? So in other words, expression of such variant U6 in a given cell would change splicing patterns potentially to all introns in that cell. And so there is more thinking and working experimental work needed to ask these questions. But that's exactly the direction that I would love to go to. Now another question.
Last question from Zofia Szwejkowska.
Okay, so here, cryo-EM seems to be confined to see only very stable conformations. Well, so you know, clearly cryo-EM is still developing, very rapidly developing, and there will be perhaps new methods, new modifications that will be targeted at those less stable transient structures. I can think of some ways that this could be done, and I know that they are being tried. So is it a question of stability, or is it a question, you know, that is notorious for any catalytic events, that they are so transient and so short-lived that basically it is impossible to detect them. So to some extent, I wonder if our molecular genetic approaches are not the most suitable to this detailed type of analysis. There is, even with improved cryo-EM techniques, it still may be difficult to capture those extremely short-lived catalytic events. But I hope I will be proven wrong, and that we will see those wonderful catalytic conformations soon.
Okay, thank you very much. These are all questions. Before I will ask President of the Polish Biochemical Society, Professor Andrzej Legocki, to finish our meeting, let me inform you that on the platform Zoom, the viewing was 100 persons, and that's the maximum license we got for Polish Biochemical Society. So, and on the Facebook streaming, we're in the peak 22 persons. So altogether 120 persons were viewing our lectures. I think that being a technical host, I'm happy with these numbers, because this was the first experiment we did. So let me ask Professor Andrzej Legocki to finish our meeting.
Well, first of all, Magda, thank you very much for your great talk. It is really a great opening of the whole series, I hope, of the lectures presented by our society. And I do hope we will announce soon several further lectures. But definitely your lectures was exceptional. Thank you very much, all the participants, for your participation as well. Thank you very much.
Thank you all again, and thank you for participation. And I'm looking forward to the rest of this great season and the new format. Thank you.